The human eye originates as a direct projection of the diencephalon, a morphogetic symphony where neurons orchestrate vision, transforming light into
The eye is an extension of the brain. Formation begins with the optic sulcus. These grooves evaginate into optic vesicles, extending toward the surface, establishing the visual apparatus.
The eye field is initiated by the master control gene PAX6. Suppression at the midline by sonic hedgehog creates disruption leads to cyclopia, where the eye field fails to divide. The optic vesicle induces the lens placode. This placode invaginates to form the lens vesicle. Simultaneously, the optic vesicle becomes a double-layered cup. Its layers become the neural retina and RPE.
The retina is not a peripheral organ. It is a specialized island of the brain. It contains all cardinal brain elements, making it a model for connectivity.
In the early stages of embryonic development, the eye begins to form. Sulci appear as shallow grooves in the forebrain. These grooves deepen and expand to optic vesicles. This is the foundation of vision. The process begins as the optic vesicle contacts the surface. This triggers a dramatic inward folding, or invagination. This double-layered cup will soon form the retina.
Initialization complete. Connection sequence pulsing. Optic nerve completion successful.
The retina relies on a complex network. Mueller cells provide architectural and metabolic support to every layer. Retinal glia and brain astrocytes manage potassium. This is specialized into potassium siphonings directly into the blood vessels, while brain astrocytes use coupled networks for spatial buffering.
Mueller cells show a high density of KIR 4.1 channels, 94% of conductance in the end foot. This siphons potassium from synaptic layers, driven by electromotive force.
Mueller cells and astrocytes collaborate. They convert toxic glutamate into non-toxic glutamine. Its loss leads to decay. KIR 4.1 is the primary potassium channel. It is highly concentrated at the astrocyte end foot. This shows its distribution across the end. This process is vital for maintaining retinal homeostasis.
Excess potassium is cleared to maintain retinal homeostasis. Richus or blood, spatial buffering K+ through the syncytium. This mechanism prevents neuronal hyperexcitability.
Glutamate is released from the neuron. Astrocytes rapidly clear it from the cleft. Glutamine synthetase converts it. Safe glutamine is recycled back. When light hits the photoceptor, it triggers a graded depolarization, closing channels through an ATP response. The retina is an active computational processor. It reformats visual information. This is necessitated by the biological bottleneck of the optic nerve. Sensory systems transform visual input. Natural images contain massive redundancy. The retina acts as a decorrelation filter, highlighting only edges and sudden motion. The retina optimizes this process, maximizing information between stimulus and response. The signal splits into 15 parallel streams to prioritize high salience features. The output of these calculations is a highly efficient, compressed neural code is partially solved. V1 then interdynamically with higher visual areas through feedback lease construction. Midget retinal ganglion cells carry high-resolution data. They project exclusively to the parvocellular layers, enabling the perception of fine detail and color. All stratified RGCs initiate the pathway. They project directly to the interlaminar KLGN. This drives the blue-yellow color response, connecting directly to blobs in the primary visual cortex. The BBB and BRB protect the CNS. The eye presents a unique, bifurcated system, composed of an inner and outer component, regulating exchange to prevent excitotoxicity.
The inner barrier has tight junctions of endothelial cells. These cells lack vesicles, restricting permeability. The outer barrier is formed by the RPE. It separates the retina from the choriocapillaris.
Research unmasked functional differences between these barriers. In the brain, P activity conceals other transporters. In the retina, however, PEEP is reduced. This means the retina has a different susceptibility.
These barriers utilize shared pathways, where deficiency leads to zonated leakage, suggesting a higher degree of intrinsic programming in the RPE. This is the blood-brain barrier. Endothelial cells form a tight seal. Claudin-5 and occludin proteins lock together. They create a highly selective protective barrier.
The inner blood-retinal barrier, or IBRB, is composed of specialized endothelial cells. Tight junctions, formed by Claudin-5 and occludin, create this impermeable seal.
The outer blood-retinal barrier is composed of retinal pigment epithelial cells. Here, tight junction proteins like Z01 and claudins form a strictly impermeable seal. The optic nerve is a CNS white matter tract. Oligodendrocytes myelinate these axons. This creates major hurdles for regeneration. Progenitor cells remain active but fail to repair. This continuous production maintains myelin length. However, this capacity does not translate into repair. Localized lesions often fail to repair. This leaves persistent areas devoid of vital glia. The optic nerve is a CNS tract. In MS, immune cells attack its myelin. This confirms it as brain white matter.
In the central nervous system, oligodendrocytes wrap multiple axons. In the peripheral nervous system, Schwann cells insulate just one, boosting nerve impulses.
Inside the sheath structure, some fibers have no neurolemma. But in the peripheral system, a neurolemma is present, formed by Schwann cells aiding repair. One oligo can myelinate 50 axons, while a Schwann cell wraps just a single axon. Central barriers are strict BBB type. Peripheral nerves use a BNB type barrier. The retina is a window to the brain. Through oculomics, we can now detect early signs of neurodegeneration non-invasively.
OCT has become the gold standard, offering high-resolution 3D volumes. Thinning of the RNFL and GCL is a proxy for global brain atrophy. The retina displays the same markers found in the Alzheimer's brain. Retinal thinning reflects neurodegeneration in key memory centers. Changes in retinal vessels provide a biomarker for dementia risk.
Parkinson's affects more than just the brain. Retinal scans reveal changes in microvasculature. This mirrors neuron loss in the brain, making the eye critical for early diagnosis.
In MS, the retina is a model for neurodegeneration. Progressive RNFL loss correlates with disability. OCT allows clinicians to monitor therapy in real time. Even in ALS, the visual pathway is involved. High-resolution imaging detects subtle nerve degeneration. Huntington's models show thinning sheaths and photoreceptor damage. AI can identify retinal signatures of disease. This predicts cardiovascular and CNS disorders. This approach transforms the routine eye exam. It is a systemic health screen. Alzheimer's pathology can be seen in the eyes. Retinal thinning and amyloid deposits are early signs. In the brain, it causes hippocampal atrophy, driven by the spread of toxic amyloid plaques.
Parkinson's leads to substantia nigra degeneration. This triggers microvascular changes in the brain. Retinal scans reveal thinning of the ganglion cell layer. These biomarkers are crucial for early diagnosis.
Multiple sclerosis attacks the central nervous system. Optic neuritis causes acute inflammation, leading to physical RNFL thinning. This reflects global white matter axonal loss.
Huntington's disease causes severe striatal atrophy. It drives profound myelin thinning and triggers irreversible RGC loss.
ALS causes the steady loss of motor neurons. Both upper and lower pathways begin to degrade. Even retinal ganglion cells show micron-level decay.
I really can't see anything. Is there supposed to be a number? It is a bright red 26. You have got to be kidding me.
In a perfect eye, the lens focuses light directly on the retina, with short-sightedness to focus in front of it. Long-sightedness occurs when a flat lens focuses light behind the retina. The shared fate of the eye and brain is encoded in their genetics. Retinal imaging allows us to map the genetic risk for brain disorders. This is the future of mapping brain health.
Retina health is tied to brain structure. Understanding axon growth provides the blueprint. AI moves medicine toward a future where the eye becomes a portal for treatment. The eye is an inseparable extension of the human brain. It is a central pillar of modern neuroscience.
Every discovery made in the eye illuminates the human mind.